Hi everyone, Nikki Hardy here, and welcome to Science Saturday, the chemistry of composting. Yet again, I am recording from my home. We had a bit of a glitch with the recording on February 7th at the library where the video didn't come out very well, and so there must have been some sort of internet issue or Zoom issue with the recording. So I'm re-recording this as true to the original presentation as I can. So this particular presentation is a continuation of something that I did with the Crow Wing County Master Gardeners. And if you want to look at the original presentation, I recommend going over to the Crow Wing County Master Gardeners YouTube channel and you'll be able to find it there. So let's get started. I hope that you are ready for a little bit of chemistry and biology. This is what this presentation is all about. We're going to break down some of the basics to composting because I believe that when we understand the processes that are taking place, it makes it easier to troubleshoot. And if we're being perfectly honest, there's a lot of stuff that can go wrong with composting. And even if you're not quite ready for a chemistry and biology talk, I do hope that you stick with me. I'm going to break this down as simple as I can. And just as a a word of encouragement is that you don't need to understand the nitty gritty. If you understand the basics, you're going to be so much better off as it pertains to caring for your own compost pile. So one of the most important things in any science field is making sure that everyone is operating with the same basic knowledge. And so for me, that means that we're going to define the words as we go. That way everyone is on the same page. So if we get started, what is compost? Compost is decayed organic matter that is used as plant fertilizer. I hope that you're asking, okay, what's organic matter? To which I would say that is a carbon-based material from a once living organism. Now, if you understand the pattern, I hope you're saying, well, what's a carbon-based material? To which I would say elementary, my dear Watson, but really it's elemental. So what we have here are the macromolecules that make up us and our food. And so we'll start out with the carbohydrates. And so what we have in the box here is a monosaccharide, which makes up our carbohydrates, and then amino acids, which make up our proteins, and then a fatty acid, which helps make up our fats. And an important thing, too, is to understand the elements that make up each one of these molecules. And so each one of those letters stand for a different thing. C is carbon, H is hydrogen, O is oxygen, and this side chain here is a whole assortment of different elements in different configurations. And one that I missed is the N, and the N is nitrogen. All right. So another important note is that carbohydrate molecule that is over on the right there is actually a simplified diagram. Over on the left, you'll notice that every single one of those points in the hexagon, with the exception of the O, is actually a carbon. And so you'll see that there's carbon molecules, and then some of those hydrogens were also left off the simplified diagram. So our carbohydrates are in fact a very carbon and hydrogen rich molecule. Alrighty. So if you take anything away from this presentation, I want you to remember that all composting techniques are microbial driven. Every single one. All right, and it takes a diverse assortment of microbes to get the job done well. So let's talk about some of those microbes. So we're just going to talk about very high level. There's different species for each one of these, but what you'll see is in the purple there, that is a yeast, and this is a larger microbe. Over by the blue arrow, we have bacteria, and then we have that green arrow, and that's our filamentous fungi. So you'll see here that this particular image is actually an image of compost, and it has so many different microbes that make up that compost or help produce the compost, which are then part of the compost, right? All right, so let's talk a little bit about why you should compost. So, why your peels deserve better. Number one, it enriches your soil and it helps support plant growth. This is some of the best stuff that your plants can grow in. Additionally, it connects us to natural cycles. And so, this is the idea that we should go out there and recycle our nutrients, right? And so, waste not. And then it also helps improve biodiversity of our soils because, believe it or not, when we have a biodiverse soil, we can help cut down on some of those pathogenic bacteria that exist that, can sometimes wipe out our tomatoes with tomato blight, for instance. And so having a very biodiverse soil helps minimize plant pathogens. All right, and so one important note here is that compost isn't even the final material. Compost is partially degraded, and it's the in-between. So when we start with an organic material, we can break it down into compost. It can be a relatively quick process, but what takes place over the long period of time is the complete breakdown into what's known as humus. So now we have another vocabulary word, and humus is the final product. It's a fully degraded product, and it is an exceptional growth material. This is the ideal growth material for just about any plant. And that's because it helps improve the soil structure, making it an ideal place for plants to grow. It also has incredible water retention abilities, not so much so that it's going to drown your plant, but it has this ability to hold on to water for the long term. And then it also improves nutrient bioavailability. And so we're not going to touch too much on that, but those would be some of our micronutrients. And so that would be like our vitamins and minerals. And so it helps make those more available to our plants because our plants are very much like us. They require certain things to grow big and strong. And then another point is that when we do good, we feel good. There is nothing better than at the end of this really great composting adventure is to put it on your plants and watch your plants grow well. There's something so satisfying to know that, hey, I did that and I'm helping that plant grow, as opposed to just getting some sort of commercial compost and throwing it out there by your plants. It's a great way to start. But I highly recommend anyone who has an interest in composting to just go ahead and do it. And then another point is that but when we compost, we can actually help reduce our landfill waste. And this is important because the USDA estimates that 30 to 40% of our food supply is in fact wasted each year within the United States. And that's important because that food that goes to the landfill is then broken down and emits quite a few greenhouse gases. So if we compost at home, we can also help reduce the greenhouse gas emissions. And another important tidbit is that gases are also produced by our microbes. And so different microbes are doing different things in different environments. So let's go on our first detour. Many people are familiar with the idea of greenhouse gases. Maybe they can name some of them, maybe they can't. But let's talk a little bit more about our greenhouse gases. So generally speaking, our greenhouse gases include things like carbon dioxide or CO2, methane or CH4, nitrous oxide or N2O, the fluorinated gases, including the hydrofluorocarbons, the perfluorocarbons, sodium hexafluoride, and nitrogen difluoride. And then I also include here CFCs because I'm a 90s kid. I grew up learning about the CFCs. They're not generally considered one of the fluorinated gases. However, it is a fluorinated gas, so I include it here. And this is the chlorofluorocarbons. And so this was one of those things that was effectively decided to be ruled out worldwide. Whether or not it was enforced and whether or not different countries did things about it is, up for debate. But in the United States here, it was significantly reduced in such a way because the CFCs were the ones that were destroying our ozone. Our ozone layer and created the ozone hole, right? And so on a high note, that is starting to heal, but that again is another detour, and we're not going to go on that detour. So let's talk about then the global warming potential, because these different gases do different things. And the global warming potential refers to the ability of a gas to trap heat relative to another gas, usually carbon dioxide, over a specific period of time, usually 100 years. So methane has 28 times the heat trapping capacity as carbon dioxide. Nitrous oxide has 265 times the heat trapping capacity, and our fluorinated gases have well over 10,000 times the heat trapping capacity. So you might be asking, well, why are we even talking about methane when there are far worse offenders? And the answer is that methane is part of our food story. So if we go and look at the greenhouse gases column over on the left side, you'll notice that I have carbon dioxide, methane, and nitrous oxide all highlighted there in green. Those are all natural within our atmosphere to a certain point. Now, it is pretty indisputable that we as humans have actually increased the abundance of many of those gases within our atmosphere. That's part of the conversation with the global warming phenomenon. But the fluorinated gases, those are not in any way, shape, or form natural to our atmosphere. Those are entirely human, man-made, and it's generally as a result of industrial practices. And so, to be perfectly honest, the fluorinated gases are huge offenders. We need to have serious conversations about it, but it is just not relevant to our food story. But methane is. So let's talk a little bit more about methane. And so with these gases here, just a quick detour or a quick note, we have a couple of new elements. And so we have F as fluorine, and then we have N as nitrogen, which I did accidentally omit on the previous slide with the amino acids. So sorry about that. And then S is sulfur. And then, of course, fluorinated gases, important note, and then we've got our natural and our man-made gases there. So let's talk about US methane in 2022. So what you'll see here is a pie chart, and we have that little tan slice of the pie that's been blown out at 17%. That is our landfills. And so number one is enteric fermentation at 27%. That's our biggest producer of methane, and that is just a natural digestive process by ruminant animals like cows, sheep, and goats, as well as the wild animals, deer, and their relatives. Number two is natural gas, and that includes both commercial and residential gas usage. And then third there is landfills. And this is where I feel like we can really do some serious good, and quickly. All right, so this is a graph of U.S. methane production over time, so starting in 1990 all the way up to 2022, and if I could draw your attention to the big, bold purple line, that is our landfill line, and you could say, what the hell, we are doing a pretty good job, right? That line is going down. Yes, we are. We are headed in a good direction. Methane production by landfills does continue to drop, but there is still some room for some serious improvement. Because after all, landfill is still number 3, and it does make up quite a bit of methane production. All right, so let's move on to landfills in and of themselves. So landfills are where bad food goes to pass gas. And that's just kind of a joke, but the process that takes place at landfills is putrefaction. And here we have another new word, vocabulary word, putrefy, which is to decay or rot and produce a foul smell. Well, that foul smell is the release of different gases. And those gases include methane, hydrogen sulfide or H2S, and that's the rotten egg smell, ammonia or NH3, and then the amine gases. And remember that composting is microbial driven. Well, putrefaction is as well. It's just a different set of microbes under a very different set of an environmental setting. So if we talk about where these different gases are coming from, you'll see that Fats and proteins can produce a lot of them, though carbohydrates can as well. So methane can be produced by fats, proteins, or carbohydrates. I just illustrate this here with fats because it's really easy to see that in the breakdown of fats, it would be really easy for that carbon there in the middle to accept two more hydrogen molecules and become CH4. And then we have hydrogen sulfide. So if I could draw your attention to the S that's highlighted there in cysteine and methionine, those are two of the amino acids that are sulfur containing amino acids, and hydrogen sulfide can readily be formed from either of them. Also, we have ammonia, which can be produced by any one of the amino acids with the N terminus of the, or with the NH2 group of the amino acid breaking away from the amino acid, producing an NH3. And then we also have the amine gases, which are highlighted there in blue. Sorry about that. And then putrefaction also produces slimes, which are just in and of themselves quite disgusting, especially when you end up with putrefaction happening within your own residential composting adventure. So in traditional composting, putrefaction happens. Got to know what you need to do to troubleshoot it, right? So putrefaction happens when, ultimately, there's not enough oxygen. And this ends up leading to uncontrolled anaerobic conditions. And I say uncontrolled because there are different times in which you do want to have the absence of oxygen. We'll talk about that at the very end with Bokashi fermentation. But in traditional composting, so I'm talking heaps and vermiculture, you need to have that oxygen present because that is the environment where microbes are doing their job the appropriate way and breaking things down without putrefaction taking place. And so putrefaction can also happen if the pile gets too wet or compacted. And this really kind of makes sense because if you drench your pile, you are pushing all of the oxygen out of your pile. And so that ultimately results in a oxygen depleted heap, right? Or if you compact it, once again, you're compacting it down and pushing all of the oxygen out. If the microbes don't have access to oxygen, they can't do their jobs appropriately. Another instance in which putrefaction happens is if you end up with an imbalance. So we're going to talk a little bit more about the carbon to nitrogen ratio later on in the slides, but we'll introduce it right here. So when we have an imbalance that takes place, that refers to we have too many food scraps, which are nitrogen rich, and too few brown sources, which are carbon rich. And hopefully right now you say, oh, nitrogen, I remember that. That refers to proteins, right? And so these are our leafy greens and the other green materials or yellow materials, any type of those recently alive materials that still have quite a bit of plant proteins available. And so one last note is that putrefaction is in fact a method for degradation. However, as it pertains to residential purposes, it's more like the gross cousin to composting. It's generally not desirable, right? All right, so let's talk about the differences between putrefaction and decomposition. And we're going to talk about this because I feel it's really important in troubleshooting what happens or what goes wrong during composting. So remember that composting is microbial driven, and so that includes the bacteria and the fungi, as well as the enzymes that are produced by bacteria and fungi. So now we have a new vocabulary word again, enzymes. And enzymes are proteins that help speed up a process. All right, so putrefaction and decomposition are dependent on what type of microbes are there and what types of enzymes are being produced by those microbes. It also is dependent on the chemical structure of the organic material being our food scraps and our brown material, right? And so any type of food or compostable material that is high in protein and fat is going to be challenging to break down. And if you think about this, if anyone out there that's watching this has had any type of composting material or composting experience or read about composting, one of the things that we don't put on our traditional heap compost piles are things that are high in protein and fat. So keep the dairy out, keep the meats out, right? So this should all kind of make sense. Proteins and fats are challenging to break down. It'll definitely swing things more towards putrefaction than it would towards decomposition. In contrast that with carbohydrates, carbohydrates are generally quite a bit easier to degrade. And for this, we're actually going to have a personal challenge. So if you have any saltine crackers in your pantry, go ahead and get one. Put it in your mouth and start chewing, and keep chewing until you got this mush, and then keep chewing some more. And what you'll notice is that over time, as that mush continues to break down, it is going to taste sweeter and sweeter. And that's because in our bodies, the first step for digestion is actually in our saliva, and that is the production and release of amylase. It's an enzyme that breaks down amylose, which is the primary starch that is present in saltine crackers. And so that is how composting works as well. And so you want this degradation of these big complex molecules into its most simple forms. And so that's what amylose does in our saliva, just like the microbes that produce enzymes do within our compost heaps. All right, so decomposition is driven in part by hydrolysis. And so this is one of the first steps to decomposition. So hydrolysis, another vocabulary word, let's break it down. Hydro refers to water, lysis is to break. So hydrolysis is one of the first things that takes place within composting. And the goal here is to break down a macromolecule into its smaller building blocks. So that means for carbohydrates, for complex carbohydrates, we break those down into monosaccharides and disaccharides, or the simple sugars. And then the proteins into amino acids. Proteins to peptides to amino acids. And then our lipids or our fats into the fatty acids and the glycerols. And so for our visual learners out there, this is just a really simple example of what hydrolysis might look like. Once again, remember, hydrolysis is going to be one of the first steps that takes place in decomposition reaction. So here we have maltose. It's a disaccharide that is composed of two glucose molecules. So you'll see two of those glucose molecules that we've referred to a couple times now. And then H2O, also known as water. And so this water and this maltose are there. And what we'll see is that we'll have that bond break, the disaccharide bond that keeps those two glucose molecules together. Those will break. You'll see that H2O has been split among those two glucose molecules, and that's it. That is how hydrolysis happens. And so we broke down a single molecule of maltose into two molecules of glucose. All right, so let's talk a little bit about go into deeper detail into hydrolysis. And so one important thing with decomposition is that it has to take place at a certain rate. If it goes too slow, we drive it towards putrefaction, because we'll end up just introducing non-ideal microbes into the mix the longer that it sits, or it's just hydrolysis is an important first step, and the rate of hydrolysis is dependent on a couple of different things, including the accessibility to bonds, as well as whether or not there's any enzymes that are present. So we talked about amylase that is present in our saliva. Well, microbes have a whole assortment of different enzymes that do different things depending on whatever it is they're trying to break down. And so here we have some of the complex carbohydrates from plants. We have amylose, which is actually a pretty simple structure. You'll see that that's just a chain of different simple sugars, right? And then we have amylopectin, which is more of that branch structure. And then we have cellulose. And cellulose, an important thing here is that you have to remember that all of these are 3D structures, and cellulose is actually kind of tube-like. And so if we think about how easy water will have a time getting into those different bonds in between those simple sugars, amylose is going to be the easiest, because there's not really anything that's preventing that water from getting into those bonds. Amylopectin is going to have a little bit harder time because of its more sophisticated structure, but cellulose is very densely packed, and cellulose is going to take the longest degrade. And so the starches within our plants are amylose and amylopectin. Cellulose is actually a structural carbohydrate and is one of the major carbohydrates in our brown material. And so hopefully this is starting to make sense. Cellulose, brown material. Cellulose takes a long time to break down. Brown material helps keep some of the structure within our compost piles. So hopefully this is all kind of connecting in a way that you go, okay, yeah, this kind of makes sense. All right, so that's kind of it for carbohydrates. So let's go ahead and move on to proteins. So hydrolysis of proteins are actually quite challenging, and that has a little bit, actually a lot, to do with the general structure of proteins. So proteins are made up of amino acids. Amino acids have this great diversity of side chains. So that's that R group that was present in the amino acid structure that I initially showed. So what we have here highlighted in blue is the unique side chain for the 20 common amino acids. Now, these are the 20 common amino acids, but there are also non-canonical amino acids that are available to be broken down as well. And each one of these amino acids has completely different properties and behaviors towards water. And we refer to this as hydrophobicity. So hydro meaning water, phobic meaning afraid of or away from. And so some of these amino acids that were highlighted in pink include valine, leucine, methionine, isoleucine, phenylalanine, and tryptophan. These are all some of our most hydrophobic amino acids, and these also play an important role in our higher order protein structure. All right. All of the amino acids play an important role in protein structure, but those ones can form some really interesting things. And so the first level of protein structure is the primary structure, and that is just the simple amino acid chain. And so if you could string together a whole bunch of those 20 amino acids that are listed, that would be the amino acid chain, which is our primary structure. Those hydrophobic amino acids play a role in the development of alpha helices, which are presented there over on the right. And then we also have a different assortment of amino acids that can form beta-pleated sheets. And so what we have is we go from this amino acid chain into these higher order structures. Well, we don't stop there because those structures can then fold in on themselves. And we also have additional reactions, or not reactions, but interactions that take place between amino acid side chains. And we also have hydrophobic interactions, disulfide bonds that can form between, say, cysteine amino acids, as well as hydrogen bonds and others. And so what we have is this full folding that's taking place. And as these proteins are folding, they're becoming more and more inaccessible to water. And then the final order of structure is the quaternary structure. And this is where we have maybe multiple subunits coming together. So two distinct proteins that come together to form a functional final protein. And so quaternary structure. So as we go from primary structure to secondary to tertiary to quaternary, it becomes more and more challenging to have water infiltrate that structure and perform hydrolysis. And so proteins are challenging to break down. That's it. That's all you need to know. Proteins are hard. All right, so proteins are bad, but believe it or not, lipids are worse. They may not be as complex as proteins with respect to their structural story, but just remember that oil and water don't mix, right? And so if you're trying to put water in these different fat molecules, it's just not going to happen. And that has a lot to do with these hydrocarbon tails, which is kind of highlighted here in pink. So we have this CH2 repeating group, right? And those hydrocarbon tails repel water molecules, and that makes hydrolysis challenging without some sort of additional input. And that includes things like emulsification, so the inclusion of different salts, as well as enzymes, in particular lipases. And so if your bacteria within your compost pile aren't producing these different salts or lipases, or these different microbes, not just bacteria, but microbes in our compost pile, they need to be producing lipases in order to have fats being broken down. And proteases for proteins, important note for the last slide. All right, so just kind of to review, we've got our decompositions. It all kind of starts with hydrolysis, and so decomposition requires water, right? We need a certain amount of water, but not too much water, otherwise we flood our compost heap and we end up pushing out all of our oxygen. Also something that we don't want to do. The goal here is to break down our macromolecules, so our big molecules like complex carbohydrates and proteins and fats, into the building blocks that make up those macromolecules. So complex carbohydrates into monosaccharides and disaccharides, and then we have our proteins into amino acids, or proteins into peptides and amino acids, and then lipids into our fatty acids and glycerol. And why go through all of this? It's because our food is made up of macromolecules. And so if you go through and you think about the different foods that we eat, the different foods that can and cannot go into our compost pile, hopefully you have a general understanding of why that is now. It's because that our foods that are high in protein and fat are just really hard to break down. That's it. And so that was a really long and in-depth build up to, there are certain foods that just do not work well in traditional aerobic composting. All right. More review. Food waste is in fact a significant problem. USDA estimates that 30 to 40% of food is wasted. Among those are some of the worst offenders are our perishable foods, and that makes sense because they're not shelf stable for a long period of time, right? And so we have overconsumption problems, we have just food waste problems, we have food being purchased and then not being used, and so we've got fruits, veggies, and then also our bread and bakery that makes up the bulk of our perishables. But also some of the worst offenders include our dairy products. They just don't have a long shelf life in comparison to some of our preserved foods. And then we also have cooked foods. So the leftovers, for instance, that sit in the fridge, those aren't the type of foods that generally work well in a compost pile. And those are also some of the food waste problem. And then when the food is sent to landfill, they go there and that food ends up putrefying because of a lack of air. And that is just by landfill design. And so landfill is one of the first step in the back of the garbage truck, right? Is compact. And so it's pushing out all of that air, and that's just by design. And when our food then putrefies, it leads to the production of our greenhouse gases, as well as some of the other more odorous gases. And another important tidbit that you learned is that compostability is dependent on macromolecular structure. Basically, some of your more complex foods are some of the foods that are just not going to work well in a traditional heap. And that is dependent on carbohydrates, proteins, and fats. Those three items can be found on our nutrition labels, and so it kind of makes a good understanding of, what types of foods are more compostable than others. And then another important tidbit that I've emphasized here over and over, especially over the last couple of slides, is that many foods have limited compostability in traditional systems. And so the question is, what can we do? And I say, we compost in multiple ways. All right, so we're going to very, very briefly touch on the different techniques because it fits so nicely together with some of the chemistry that I presented here. And so our residential techniques that I'm going to introduce here are heaps, worms, and pickles. And before we get into that, an important reminder that all composting techniques are microbial driven. Remember that, all of them microbial driven. So heaps, worms, and pickles. These are all dependent on the bacteria and fungi, as well as the enzymes that those bacteria and fungi produce. It is also dependent on the surface area of the food molecule itself. And so smaller particles are much easier to break down, with the caveat being that smaller molecules are more likely to compact as well. And so there's a bit of a trade-off there. And another important concept to understand is this idea of aerobic. And so aerobic digestion takes place in an environment with air. Aerobic means with air. And this includes things like heaps, which are traditional compost techniques, where you just might pile it on in the backyard, right? Or it could be a tumbling composter. Maybe you have a pallet bin. Those are all different types of heaps. And then there's also vermiculture, which is a aerobic technique that uses worms to help move the digestion along faster. In both of those techniques, it requires air, which means that there needs to be some sort of additional input put in so that the compost heap is turned in some way, shape, or form to introduce air into the heap at different periods of time. And that's important to prevent compaction as well as putrefaction. And so with heaps, that means that, you, me, or maybe my kids, we got to go out to the heap, we got to flip it over, make sure that the air is circulating throughout the heap. With vermiculture, it's a little bit easier. The worms are doing the job for you. And then we have anaerobic techniques, and these are the techniques that are done without air, an meaning without, aerobic, air. So anaerobic is without air. And the big one for residential is Bokashi, and this is pickling your food scraps. And this is kind of a fun one that's relatively new to me, and thankfully I've done with quite a bit of success this past winter in particular. And Bokashi requires a special combination of facultative microbes. And facultative microbes are ones that can operate with air or without air, but they have different types of biological processes depending on the environment. And so in a lack of air, so without air, they switch to fermentation, which is really cool. And so that refers to the pickling process, right? But then when they are exposed to air, they continue to exist and thrive, but they switch into general respiration. And so these facultative microbes can play a really awesome role in both aerobic and anaerobic decomposition of food scraps. And one more thing worth discussing is that brown and green sources. So I've kind of alluded to brown and green sources being our food scraps, brown sources being more structural. So let's dive a little bit deeper into what those are. So green refers to the nitrogen rich sources. So nitrogen, hopefully you say, aha, I remember that, that's proteins. And these green materials are essential for bacterial reproduction as well as enzyme production. And so hopefully that all makes sense. In order to have bacteria reproduce, it is a protein intensive process, and enzymes themselves are proteins. And so it makes sense that we need nitrogen rich sources to produce the proteins that are required for bacteria to reproduce, as well as for the enzymes to be produced to help break down those food scraps. All right, and then we have brown. So these brown materials are carbon-rich sources, and this is the lignocellulosic biomass. And so this includes things like cellulose, hemicellulose, and lignin. So hopefully you're saying, oh, I remember cellulose. That's that really compact structural carbohydrate from plants, right? Well, hemicellulose and lignin are as well. And so what we have here, because this is a chemistry talk, we have some structures here. And so this is cellulose up on top, which is made up primarily of D-glucose. Hemicellulose is in the middle, which is made up of the monosaccharides, as well as the uronic acids. And then we have lignin down at the bottom, and this is the phenol derivatives. And so hopefully you see a pattern here. These are all ring structures, right? And so these are all different materials that actually take quite a long time for hydrolysis to happen and for things to really start breaking down. And so these are long-lasting materials, and they provide a rich source of structure within our compost heaps. And so carbon is essential for all of the biomolecules. If you remember, we had carbohydrates, proteins, and fats are all carbon rich, right? So we need a lot of carbon in order to make any of these, the carbohydrates, the proteins, or the fats. And then, like I had mentioned, these brown materials also provide structure to our heap, which helps improve the aeration. It also provides energy for the microbes, and it can also act like a sponge. So when we dampen down our compost pile, any type of excess water ideally will run through, but the water will be taken up by that brown material and then released over time. Because remember, hydrolysis is one of those first steps in decomposition of our compost. Even though it's microbial driven, hydrolysis is one of the first steps. It requires water, so we need to continue to have water accessible within our decomposition heaps, or our compost heaps. All right, and then our heaps and worms, those two techniques in particular have a very specific ratio that is ideal for the decomposition process to take place, and that is 25 parts carbon rich material to 1 part nitrogen rich material. Now that seems like a lot, and it is. And so one of the questions then becomes, okay, if I have, like 1 pound of food scrap, where am I going to find an equivalent or the correct amount for carbon, right? It might not be 25 pounds, but you're going to need a lot of carbon material, right? Well, carbon material isn't actually all that hard to find. And so one of my personal favorites is dry leaves. These are super duper abundant. So what we do at our house is we leave all of the leaves down. We don't mulch them. We just let them sit. Well, my husband mulches them a bit until I tell him to stop. And the benefit to that is that it provides a really great place for pollinators to live over the winter. And so that's number one. And then number two, in the spring then, after the pollinators emerge, that's when we can start raking them up and dry them out, put them in a pile, and then all of a sudden they're really easily available for the compost pile. Another really great source of carbon is shredded paper. And so a note about our recycling infrastructure here in Crow Wing County is that it actually is not well equipped to handle shredded paper. So you might want to check if you're not in Crow Wing County, if you're someplace else. Shredded paper is not well handled at our particular facility because it falls through the sorting machine. And that ultimately makes shredded paper non-recyclable unless it's properly bagged. And so one of the best ways that you can then use that shredded paper is within your compost pile. And so if it's questionable on whether or not it's going to be able to be recycled at the recycling center, just pop it right into your recycling or into your compost pile. And then also there's cardboard. This is very high in carbon. It's great for structure, though you have to be careful to avoid the waxed varieties, and so the ones that have the shiny surfaces. There's also concerns about PFAS contamination with some cardboards, and so just be smart about your cardboard use. And then one of my personal favorites, we have 3 kids. Our kids are messy. We go through paper towels. It always makes me feel bad. However, it makes me feel less bad now that we're composting all of those paper towels and napkins. And we go through so many of those paper towels, and they do really make a great addition to the compost pile. So, it's pretty easy to find some of those greens and browns. You can find a list like this just about anywhere online. But it's also important to take note of the things that you shouldn't compost, right? And so those include things like cooked foods, cheese and dairy, meat and bones, pet waste, used tissues, produce stickers, oils and greases, glossy coated paper, so on and so forth. Different things in your compost pile. If you wouldn't want to eat a plant that grew from that, don't put it in your compost pile, right? And we just talked about used tissues and paper towels. So the cautionary statement with used tissues is just like a no for me. But with paper towels, if you do end up using that within your compost system, know that you may be inviting critters in. Because those critters will be drawn to the food waste that might have been present on those paper towels. And then I've highlighted these 4 here, the cooked foods, cheese and dairy, meat and bones, and then those paper towels, because those in particular make for a really great addition to Bokashi fermentation. And so those are things that are not traditionally compostable and in aerated systems, so like in our compost heaps or with our worms. However, it is something that we can pickle and then feed back to our worms or bury in the ground and actually use that as compost for our plants. All right, so with that, I'm actually going to stop because next week we're going to dive a little bit deeper into the different composting techniques. So it's kind of going to be a blended presentation between the chemistry of composting today and then composting techniques next week. So just as a sneak peek as to what we're going to talk about, over on the left we have this is the bin that we use at my home. It's a rolling bin. So that's a traditional composting technique. In the middle is the vermiculture bin, so you can see that that's a stacked system where the worms work from the bottom up to the top. And then over on the right, that's our pickling system or our Bokashi bin. And so I hope that you tune in next week and learn a little bit more about home composting techniques. So with that, I hope you have a super duper day and see you soon.